Thanks, David. Thanks very much for the introduction, and thank you to everybody that's put this webinar together. Thanks, everybody, for taking the time to dial in and listen. And I'm excited to tell you about Arovella Therapeutics. As David just mentioned, we are a biotechnology company now, pretty much focused on oncology. And for those of you that don't know us, our ticker is ALA. Next slide, please. And I will be making forward-looking statements, so please do take note of the disclaimer. And just jumping into the highlights of the company. So what we're working on is termed an invariant natural killer T cell, or iNKT cell for short. And it's a cell therapy platform that we're developing for the treatment of initially blood cancers, but looking to expand also into solid tumors. And I'll unpack what that means more in a moment. Our lead product is termed ELA-101, and it's being developed for what we call CD19 expressing blood cancers or lymphomas. And we're expecting to take that into clinic in 2024, which will be an exciting milestone for the company. Now, the cell therapy sector has been a really exciting space to consider the next pillar of oncology therapeutics, but it has been hampered by some challenges. And we believe that our iNKT cell platform is well positioned to help tackle some of those challenges and push the whole field forward. Now, we've developed a very strong leadership group with a lot of experience right the way from the research and development phase all the way through to commercialization. And there's a heavy emphasis within the team to develop cell therapies, which is quite specific and unique. In terms of the company's DNA, we have essentially worked hard to pivot the company in doing so, bringing in what we consider to be the most high likelihood of success IP from a range of institutions and universities worldwide. So we've got our lead platform from Imperial College London, and we've since acquired technologies from the MD Anderson Cancer Center, and also have an option agreement for another technology from the University of North Carolina. And we'll continue to do that where we see that there's a good fit for some of these technologies to fit with our current platform. So we are the only iNKT cell company in Australia and one of a handful globally. So we do see that we have a unique value proposition and that we are well positioned for success. Next slide, please. So I'd like to quickly unpack some of our financials. These dates are towards the end of July. So we're somewhere in the range of 40 to 45 million in terms of our market capitalization. Our pro forma cash, including the proceeds from an SPP that was closed at the beginning of July, was about just under 7.4 million. And of course, that does not include our R&D tax rebate, which we're expecting to bring in later in the next quarter, or in this quarter, I should say. And what we're very happy with our current register. We're delighted to have Merchant Group as our largest shareholder, but also equally delighted to have a number of other smaller institutions and high net worths that are quite supportive of the company in terms of its drug development efforts. So we've had a good start to 2023 off the back of presenting some very exciting data demonstrating what we're trying to embark upon and take into clinic at the American Association for Cancer Research in April. We had quite a spike in the share price. Look, it has since come off a little bit since then, but we are still excited for some of the news flow and the milestones that we're expecting to hit towards the end of the year, that we're going to continue to deliver shareholder value to all of our shareholders. Next slide, please. Now, I just want to unpack exactly what it is we do, and apologies to those of you that already know this space well. But what I'll do is I'll start with CAR T, because it's the most well-known cell therapy. And where the T comes from, just very briefly, it relates to an immune cell in our bodies. It's naturally occurring all the time, and they are called T cells. And their job is to find and destroy or eliminate anything that doesn't belong, whether it be an infection, in some case cancer cells as well. I don't have a pointer, but moving to the diagram, the way this actually works as a therapeutic for cancer treatment is we actually take the blood of the patient, and I'd like to make that very clear. It is from the patient that actually has in this case, the lymphoma. We then isolate their T cells, the immune cell. They get shipped off to a lab to be genetically reprogrammed so that they actually have, and you can see just on the right in the middle, it's called a chimeric antigen receptor, or CAR for short, which is where we get CAR T. And I'd like you to think of that as a laser-guided missile that now will tell that immune cell to go and destroy the cancer cells that have a particular marker on their surface. So they get grown up again in the lab into the millions or billions, and then ultimately they end up going back into that patient where they originally came from. And you can see on the surface of the cancer cell, there's little green bumps. The newly engineered T cell will find those bumps, and as soon as it does that, it will trigger the cancer cell's destruction. So just to make it clear, we start with the patient's material and we give it back to the patient. Next slide, please. So this has, as I mentioned a moment ago, it has revolutionized the way we think about cancer therapeutics, because we are now at a position where we are starting to use the word cure and cancer in the same sentence. And this is off the back of a series of media reports coming out after some research demonstrated that patients that had taken the therapies both in the adult setting and the pediatric setting, they were now 10 years cancer-free. So that's last year, so it's even beyond, which is quite exciting. Accordingly, the first two approved products have done quite well in terms of revenue. And naturally, with a market like this for oncology, it is expected to exceed more than $60 billion by 2030. So it's set to be a very important pillar, both from a healthcare perspective, but also commercially. Next slide, please. Now, as I mentioned earlier, there are a number of challenges that do still hamper the therapeutic class as it stands today. And I mentioned a moment ago that T cells that are used in this process, they must actually originate from the patient. And what this means is that for every single therapeutic dose that's manufactured, it needs to be manufactured bespoke. And the pharmaceutical industry, like many industries, wasn't necessarily set up to be making a therapy for every single individual. So what this results in is quite a complex supply chain, and naturally a very high manufacturing cost, which leads to the therapy costing more than $500,000 per dose per patient. So also we know that because the starting material comes from the patient, it's likely that the immune cells themselves are compromised due to the disease or due to certain treatments like chemotherapies that can actually reduce the fitness of the immune cells. In addition, not all manufacturing or cancer centers are actually able to manufacture these types of therapies. So there will be a number of eligible patients that naturally will not be able to be treated with those particular CAR-T products. So in addition, it takes about 4 to 6 weeks to get the therapy manufactured for each patient. And unfortunately, some patients will not make that journey. They'll actually die before the therapy can be manufactured. And of course, because we're continuously making it for every single person, there's a high likelihood, well, not a high likelihood, but there's always the likelihood of a manufacturing run failure, which again increases time and costs. So we do believe that our iNKT cell platform addresses these manufacturing and logistics challenges, but we also believe it has the potential for improved efficacy over what's already available. Next slide, please. So I would like to briefly introduce the invariant natural killer T cells, or iNKT cells for short, just so I can contextualize why we're so interested in them. And what you can see in the top right corner is the T cell, which is where most or all approved CAR-T products sit. Bottom left is the NK cell, which is also quite a well-known cell type that people are using in this space. If you just click on the, click forward again, please. But as I said, we're focusing on the iNKT cell. And the reasons we're doing that are they actually are the front line of the human immune system. They bridge the two halves of our immune system, which we call the innate, the fast acting, and the adaptive, which is the more slow but prolonged response. They actually contain the ability or the killing mechanisms from both T cells and natural killer cells. And we know that they actually naturally target certain cancers that also already produce CD1D. Now, they also have a very strong safety profile. They don't cause what is referred to as graft versus host disease, which is what enables us to be able to give iNKT cells from a healthy individual to somebody that has cancer. So we can actually use these for, in one sense, a one-to-many approach, or what we like to call off the shelf. Now, in addition, unlike some of the other immune cell types, they also have multiple anti-cancer properties. They shape what we call the tumor microenvironment, which is the area surrounding the tumor, by actually influencing and killing certain other immune cells that essentially help the cancer cells to grow and evade the immune system. They also are able to actually infiltrate inside the tumors and then secrete what we call signaling molecules or cytokines that actually then activate other components of the immune system, like T cells and natural killer cells. Next slide, please. So we believe very strongly that the potential of CAR-iNKT cells is untapped. And if you could tap forward twice, that would be terrific. Once more, thank you, perfect. And this is just a snapshot of the players that are currently in the T cell and NK cell space. So I think you can appreciate that there's quite a lot of activity in each of these sectors. And as I said, this is a subset of some of these players. If we click once more, we'll see that in the iNK T cell space, there's really a handful of groups working on the invariant natural killer T cells. So we're quite excited because we see that we're in the leading edge of cancer therapeutics in the cell therapy space, but we've also carved out a very nice position to be in a niche area on top of that as well, which is nice. Next slide, please. Now, just to give you an idea of the workflow, so I showed you what it looks like for CAR-T previously. It is quite similar. There are two major differences that I'd like to point out. So on the very top left, we collect the blood from a healthy donor. So that'll be someone with a fully functional immune system. We then isolate their iNKT cells. We take them again to a lab like we would in the previous example, where we engineer them to produce the chimeric antigen receptor or laser-guided missile, which you can see sticking up in green. And then they get grown up and expanded into the billions in our case, so that we've got many cells, then we can store them and vial them, put them in little vials, store them in the freezer. And when a patient is actually eligible to take that therapy, it can be pulled out of the freezer and then given to that patient as and when needed. So what that means in terms of advantages is we have a healthier starting material because it's coming from a healthy donor. We can scale up to have multiple doses per batch, and it's much faster access to treatment for patients because we don't have to wait for the therapy to be manufactured. And it does remove the risk of manufacturing on failure because it's sitting in a freezer waiting to be administered. Next slide, please. And so when we look at what we're actually developing, and I haven't had time to go through each of these in detail, but our lead product is ALA-101, which is targeting lymphomas. You can see on the very left-hand side, and I'll do my best to describe this without a pointer, but we've got in the middle up the top, we're combining ALA-101 or our CD19 targeting program with another well-known biotechnology company, Immunogene's onCARlytics virus. That's a very neat program where their virus will actually tag solid tumors with the target that our iNKT cells will recognize. So we're excited to develop that to tackle a range of solid tumors. We did get promising in vitro data for that, or test tube data for that earlier this year, and looking forward to getting animal data before the end of this year, which is exciting for that program. And on the very right, we've got another program from the MD Anderson Cancer Center, which targets a unique target on the surface of, and the data suggests on the surface of multiple myeloma and a range of solid tumors. And in the very low dark blue line, we've got our cytokine technology from the University of North Carolina, which we think will enable us to increase the effectiveness of our iNKT cell platform across a number of different solid tumors as well. So really importantly, we can continue to look for new CARs to add to the platform, which is quite exciting, as there are a number that we can evaluate both in the blood cancer setting or the solid tumor setting. Next slide, please. So in terms of the next 12 to 18 months, we do have quite a busy schedule in front of us, which is exciting. Our major goal is to take ALA-101 into its first in human trial in 2024. Now, for those of many of you will know this already, but this will be done directly in patients. Nevertheless, this is considered a safety study, so we'll be looking to make sure that we don't see any dose-limiting toxicities or anything of that nature. But of course, we'll be looking to set that trial up so that we're looking for early efficacy signals of the product as well. And we'll also continue to assess novel complementary technologies, as I mentioned, that we believe enhance the platform and start to push us more towards the solid tumor space as well. So for the second half of this year, we'll complete process optimization and scale up to prepare for manufacturing cGMP or clinical grade material for clinical trials. One of the components, which is called a lentiviral vector, will have finished before the end of the year, which is a really important milestone for manufacturing these types of therapies, and we'll be finalizing our clinical trial plan. And so for the ALA-101 for this first half next year, the intention is to complete the manufacture of the product, phase 1 clinical trials, and then complete the regulatory submissions so that we're ready to start phase 1, which is a very exciting milestone for the company. And so in terms of our earlier pipeline programs, as I mentioned, we're working towards getting data for the combination of our ALA-101 with Immunogene's onCARlytics platform in solid tumor animal models. We are analysing additional CARs to add to the platform. And we shared some data recently about the cytokine technology that is currently under option, and pending our due diligence, we'll look to license that into the company. And so for those studies in the first half of next year, we're excited that we'll continue to generate proof of concept studies for those particular therapies. And again, looking towards moving the platform more towards solid tumours. Next slide, please. I thought this is a really useful slide just to contextualise some of the activity that's transpired in the cell therapy space. And I think we can all agree over the last 24 months, it's been a pretty tough sector in terms of capital raising, share prices and M&A activity. But what we have seen specifically in the cell therapy space is there still have been a number of, whether it be acquisitions or collaborations or strategic partnerships that have transpired. And importantly for Arovella, and I've deliberately left it at this list, but it is also in companies or technologies that are at quite early stages, early as preclinical, which is where we currently sit, but also a lot at phase 1, which is the next phase for us to enter. And some of these deals are quite sizable, in the order of hundreds of millions upfront payments. Again, putting that in context, that Arovella's current market capitalization is between 40 to 45 million. It's clear that this is still a sector where there's a lot of value to be created. And of course, looking for a strategic partner to get involved is also something that we're considering and very mindful of. Next slide, please. With that, I'd just like to say thanks again to everybody. Thanks, David, for moderating, and thank you to everybody for listening. I'd be delighted to answer any questions.
Thanks, Michael. A great presentation and clearly a sizable market you're operating in. Similar to the question I asked of Liz, is this something you've always been interested in? Has your clinical and your own research work led you to this point, or have you sort of cottoned onto it later in your career?
It's an excellent question, and my career's probably been cut up into multiple stages. So my first career was as an academic researcher, and not too dissimilar to Liz, actually, and a biochemist. And I think that really sets the foundation for these technologies. They all operate off an intimate understanding of biochemistry, how cells behave, how these things can actually be utilised or weaponised in the fight against cancer. And then I moved into drug development, followed by some time at a venture capital fund in Australia, and was very aware of the CAR T space, probably quite some time ago, probably if not somewhere around the 10-year mark ago. So it was a sector that I was always very fond of. And upon joining Arovella, I thought it was a very good idea to be looking at technologies in this space. And one of the key factors for us, and that slide I mentioned earlier, where there's a lot of activity for both T cells and NK cells, was how do you enter this and not become a me too? And I think we managed to do that by working on the invariant natural killer T cell platform. And it was quite remarkable, actually. I think a month before we licensed in this technology, another iNKT cell company was bought out for 185 million. Another one came out of stealth and raised 52 million and didn't list, but they raised 52 million in the US, and since ended up in an $875 million partnership. So it's clearly a sector, I think, that's got a lot of potential. Once we start coming out the other end of this pretty prolonged biotech downturn, I think we're going to see some strong value creation for our shareholders.
In terms of the iNKT product, what was the genesis? And apologies if you did talk to it earlier on, but where did it come from? What was the genesis? What was the driver?
Yeah, I didn't really delve into it, so it's a good question. So the technology was founded or discovered, I guess, at the Imperial College London, and that was an important part of our criteria, that we'd look really for top tier either universities or research institutes. So for those of you that don't know Imperial, it's consistently ranked the top 10 university worldwide. And when we were looking for a range of technologies, and we would have looked through, I think, close to 100 or more, this is one that really stood out to us for all the right reasons, right? That it was differentiated. It wasn't a very important sector. And importantly, the inventor himself, he'd spent his career researching and understanding iNKT cells. So that's not something that you can just sort of walk into, I think, and sort of fake. So that was really important. He's also a medical doctor, so he continues to treat cancer patients and sees firsthand the failures, what we're not getting right. And I think that was his driver to get involved I really firstly in having a project that was far more translational. And then when we came and approached him to license his technology, I think it was just a really good fit and a good relationship from the outset that we were the right group to develop his technology and take it into clinic. So again, I think we're all pulling in the same direction from that point of view, and it is a very exciting time taking this into clinic next year.
And you showed the slide before where there was lots of people in this area and lots of people in this area, but when you looked at the iNKT cell market, it wasn't busy. Why do you think that is? And is that the opportunity that it isn't busy? Everyone's already over in other areas focusing on other things, but there's a bigger opportunity here.
We think so. And there's a number of reasons why, as I said, the T-cell space became quite saturated. There was 2 acquisitions in 2018. One was both companies were in phase 2. One was 11.9 billion. The other one was 9 billion. And it essentially set the market on fire in terms of what could be achieved. And then natural killer cells were the next touted therapy because they can be used off the shelf or given from healthy donor to patient. T cells can't be given in that way. And so we see that as a real limitation on continuing progress. There's often media reports or fierce biotech reports that companies, the biggest pharma companies you can think of are struggling with manufacturing because it needs to be made for every single person that wants to take the therapy. Conversely, the NK cells, I think they've had trouble demonstrating the prolonged persistence in the human setting to have the durable responses that we would like to see from therapies of this nature. So that's where iNKT cells, having the good properties from both T cells and NK cells, actually fit like almost, I think another company's referred to them as the Goldilocks immune cell. Now, I think what has hampered, and we presented this recently at BioShares actually, what's hampered the uptake in the sector is they were discovered, I think 20 or 25 years plus after people were already looking at the cytotoxicity of T cells. So they have come to the fore, I guess, or to the front line quite a bit later. And it wasn't only until mid 2010s that we had the first CAR iNKT cell publications actually put out in the scientific community. So they've been a far less well-studied immune cell type. And we also know that they're lower in number in the human immune system, right? So when we actually take the blood from a healthy patient, we start with a lower amount of cells, which will make manufacturing more challenging. But what we licensed from Imperial was actually patents to manufacture the CAR iNKT cells. So I think we sit in a very good spot. We don't know the exact reasons why the space isn't busier, but for our intents and purposes, right, we're quite happy to be in that space with relatively few competitors.
And final question, you did show a slide which showed a lot of the transactions that have been occurring in the space. Now, again, I don't want to put you on the spot, and we all understand materiality and the need to disclose at the appropriate time. But when do people start talking to you? When do people start looking and going, hang on, you're doing something that is different to what others are doing, but clearly has a significant application, albeit a little bit behind from a clinical point of view in terms of where you are in the cycle. But when do they start looking at you and going, well, we see what you're doing, we see the work that's been done, the partnerships are strong, you're moving into that phase where we now start getting interested?
I think it's now, to be honest, David, that's when you start having the discussions. And look, when we presented the data at AACR in April, that did generate interest, which was great for us, right? We're delighted to have that happen. But the reality is it has happened for other companies even earlier than where we are. And I gave that example before. Appia Bio came out of stealth in May 21, raised 52 million. And then by August 21, they were in an $875 million partnership with Kite Pharma. So they hadn't reached clinic at that point. They were 2.5 years away from clinic. They're still not in clinic. So, that preclinical window, it can be large. And it seems as though for the right technology, the right pharmaceutical company is willing to get involved. Naturally, I do think some of the more sizable acquisitions that have transpired, they have been in phase 2, but we've still seen a lot of activity in phase 1, which is the next step for us. So we're quite excited, I think, by what we're developing. And again, bringing in synergistic technologies that enhance our platform, and also making sure we secure IP around them and block out our competitors in the iNKT cell space. We think that just sets us up for success, and obviously sets us up to have some pretty interesting discussions as we move forward.
On that note, Michael, thanks for the presentation. We look forward to following the company with interest. Clearly a pivotal time for the company and its shareholders. We wish you all the best and look forward to catching up with you soon.
Thank you, David. Appreciate it.